Okay, first I want to talk a little bit about how we organize our Streptococcus. For instance, in your book, they refer to Strep pyogenes as GABHS. Okay, what in the world does that mean? Well, I want to tell you. Okay, so when we categorize our streps, okay, we use two different schemes that are really don't have anything to do with each other. Okay, the first way to categorize them is by Lancefield groups. Okay, and this is a grouping system. It was named for Rebecca Lancefield. She was a researcher, 1920s, I believe. And she discovered that most of the streps have these sugars on the outside of the cell. Okay, and that she could split up the streps based on what kinds of these carbohydrates they had. Okay, so she put the streps into groups based on carbohydrate surface antigens. Okay, in lecture you're going to talk much more about what an antigen is, but an antigen is just something that elicits an immune response. Okay, so she could use antibodies in order to very specifically figure out, what kind of sugars were on there. Okay, so with this grouping system, she puts certain organisms in group A, some in group B. I think there's a group D, E, all the way down to, I think, at least J. It might go lower than that, but it skips some letters, so I'm not sure why. Okay, so there's maybe between 15 or 20 of these Lancefield groups. Some streps don't have these surface antigens, so they don't fall into this grouping system, so that makes it a little bit not perfect in my mind, but it's still useful. For instance, our strep pyogenes here, this GAB, that stands for group A. Okay, so strep pyogenes is in that group A. Okay, and some of the groups only have one organism. I think group B is only Strep agalactiae. Some of the groups have 3, 4, 5, 6, 7 different species in it. Okay, so the other way that we're going to rely more on in lab, because we can actually test that, using antibodies is a little bit beyond what we're going to do in introductory lab here. We're going to categorize streps based on their hemolysis patterns. Okay, so these two different ways to categorize streps are totally independent from each other. Okay, and not necessarily having anything to do with the clinical significance of the organism. Okay, so we can categorize our streps based on their hemolysis patterns when they're streaked out on blood agar. Okay, so do they have a characteristic way that they lyse the red blood cells? Okay, so there's 3 different hemolysis patterns. Beta hemolysis is characterized by total red blood cell lysis. Okay, so when we streak our beta hemolytic organisms out on a plate, we get kind of a yellow clearing in the media, that red media. Okay, I'll show you a picture of that in a minute. Okay, strep pyogenes is a beta hemolytic. So this is where the acronym comes in here. Group A, beta hemolytic strep. Okay. Now, organisms that are beta hemolytic they're beta hemolytic because they have one or two different hemolysins. Okay, if they have the O hemolysin, that means that that hemolysin is inhibited by oxygen. And if they have the S hemolysin, that means that that enzyme is stable in oxygen. Okay, and I bring that up because that has repercussions as to how you're going to streak your organism today. Okay, so with your assigned strep, you are going to take some blood agar. Now, is this blood agar selective, differential, neither, both? This is just regular blood agar. There's no antibiotics in there, so it's not selective. It is differential because as you'll see, you'll be able to discern the different hemolysis patterns. So certain organisms look different than others, so that tells us something about the organism. Okay, so when you're streaking your organism out here on your blood agar today, I want you to make the first quadrant maybe a little bit bigger than normal, and make sure you get a good heavy inoculum on there. The streps are a little bit harder to grow, so you want to make sure that you get plenty of organism on there. Maybe even do a double inoculation. So flame your loop, dip back into your stock, and streak that first quadrant again, just to make sure we get good growth there. Okay, then you're going to flame your loop and do your 4-quadrant technique like you would normally. Want to make sure you get lots of isolated colonies to make sure that, because when we have isolated colonies, that helps us discern and make sure that we have a pure culture. Okay, so flame your loop, streak it out, flame your loop. After you streak that 4th quadrant, try to use up all your surface area, and before you flame your loop, I want you to go back to the 1st quadrant and stab down in that agar, maybe 3 or 4 times. Okay, we don't usually get to do that, so poke away. The reason that we do that, and it's very important that you don't forget to do that, because in the stabs, there's going to be less oxygen down in there. Okay, so we're not worried about the organism growing. Remember, the streps, most of them don't really care whether they have oxygen or not. So it's not to get enhanced growth, but we want to see enhanced beta hemolysis. Because if your organism is considered a beta hemolytic only because they have this O hemolysin, you're only going to see that beta hemolysis around the stabs where there's less oxygen, and you won't see it anywhere else. So one of our organisms only has that O hemolysin. One of our beta hemolytics actually has both hemolysins, so you're going to see that clearing of that beta hemolysis everywhere there's growth. Okay, so this again dictates how you're going to streak out your organism. Okay, so 2 of our organisms are beta hemolytic. 2 of them are considered alpha hemolytic. And alpha hemolysis is characterized by partial red blood cell lysis. So essentially how that looks around the growth, that blood agar generally turns kind of a dark greenish, brownish color. Okay, so I hope you're thinking, wait a minute, how can a red blood cell be partially lysed? Seems like either it's broke open or it's not. But organisms that are alpha hemolytic, they lyse open the red blood cell, but they don't degrade the hemoglobin. Okay, the hemoglobin stays intact, and that's where this kind of greenish brownish color comes in. Okay, so we have 2 organisms that are alpha, 2 that are beta, and we have 1 organism that's considered a gamma hemolytic, and that's just characterized by no red blood cell lysis. Okay, so essentially we get no change in the media around a colony that's gamma hemolytic. You could scoop that colony up off the media and hopefully the media wouldn't look any different. Okay, so here's an example of the different hemolysis types. Okay, right, the beta hemolytic here, that's really obvious, especially when you hold your plate up to some backlight. Okay, hold it up in front of the window, hold it up to the light source. Okay, that light should shine right through there. You could maybe even read through there if you needed to. So that's obvious as long as you don't leave your plate sitting on your bench. That's when students miss it. Again, it may only be around the stabs. Okay. The alpha and the gamma are a little bit harder to discern between, and this isn't the greatest picture. This is one of those things that you're going to have to spend some time looking at other people's plates to learn to discern between an alpha, a beta, and a gamma hemolytic. Sometimes the alpha and the gammas will be a little bit darker around the stabs. I think sometimes the air gets in there and it dries the media out a little bit more. But typically, you would never have an organism that's, oh, beta in one place and alpha in another. If it's only one organism on there, it's, we label it as either an alpha, a beta, or a gamma, okay? Not a couple different things. Okay, but we can't tell our 5 different organisms apart based only on their hemolysis patterns. Like I said, we have 2 betas, 2 alphas, and 2 gammas. So we have a couple of discs that we're going to throw on here on the right-hand portion of our first quadrant. And that will, and the organism's sensitivity or resistance to those discs will help us discern, along with hemolysis patterns, what organism we have. All right, so we can tell all 5 of our streps apart based on this one blood agar plate. Okay, I ran out of room there, so I got to write down here. Those discs, okay, one of them is called an opticin disc. And opticin is actually not an antibiotic, but it's a chemical that some of our streps are sensitive to, and some are resistant. And this is especially helpful in discerning between our alpha hemolytics. Okay, so that opticin disc is actually abbreviated a P disc, and the other is a bacitracin disc. And that's helpful to tell our 2 betas apart. One of our betas is sensitive, one is resistant, and we call this an A disc. All right, so not intuitive, it's the second letter of the name instead of the first. These are in some small glass vials, and make sure, again, you're always reading the label on there as to what's what. If you remember back when we did the Kirby-Bauer test, bacitracin was one of the antibiotics we tested in that Kirby-Bauer test. Only when we did Kirby-Bauer, we had a B10 disc that was a bacitracin disc. Make sure that when you're testing Streptococcus, you don't use a B10 disc, because that's a different concentration of the antibiotic, and it will give you the wrong result. I forget which is more concentrated and which is more dilute, but you want to make sure you use that A disc because, again, it's a different concentration. Okay, so we don't give you too much room here. You don't want to put these 2 discs right on top of each other. Remember, if you didn't know what organism you were streaking out, you would just go ahead and put both those discs on there. You never want to have a plate that's already grown up and then put the discs on. That's not going to do any good. Okay, so you got to put them on when you first inoculate the plate, And again, you're really only looking for a really small zone around there, so you can have them pretty close together there and still give you the information that you need. Okay, so that's the plate that you're going to inoculate. Okay, and that's what we have for lab 14. Okay, lab 15 is really just some confirmation tests. Like I said before, you should be able to identify your strep based on that one plate, that one blood agar plate. But again, the hemolysis can be a little bit tricky to read, and so it's kind of helpful to have a couple other tests. Again, may or may not be helpful. The bioelasculin test is positive for group D strep. And I didn't really talk about which of our streps was group D. It's actually the Enterococcus is in that Lancefield group D. So if you suspect that you have Enterococcus, by all means inoculate a bioelasculin test and that will tell you for sure. This one's easy to read. This is a slant. Esculin is actually a polysaccharide. And some organisms, some of our streps, only a few of them, can break down esculin to esculatin and glucose. So basically it just cleaves off one glucose unit and you're left with a slightly smaller polysaccharide. I'm not going to worry about the enzyme here that these organisms use. I've heard it called a couple different things, and your book doesn't mention it. So esculinase is probably the most common thing it's called, but don't worry about it. Most organisms can't break down esculin, especially in the presence of bile. This is really a selective agent here. The bile, it's a detergent. Most organisms can't grow in its presence. So these group D streps are kind of unique that way. Okay, so this is what the bioelasculin, we usually label these BEA, bioelasculin agar. This is what it looks like when it hasn't been inoculated. Remember, when you're inoculating a slant, with your loop, you're just going to streak on the surface there. Okay, we'll incubate it. And there's actually some iron in that slant that will react with the esculatin to give us a black color change if the organism is positive. All right, so you don't need to add any reagents in there. It's already in the media. And after incubation, we get a nice black color change. So this one is really very obvious and easy to read. The next test, the sodium hiparate test, is a little bit, I would say it's not quite as reliable. Definitely. This is one that's positive for group B streps. And like I mentioned in the last video, that's Strep agalactiae. That's our group B strep. Let me just take a minute here because I think it's helpful to kind of remember what's going on to draw out this structure. I don't have it memorized, so wouldn't want you to worry about that either. But it is a little bit useful to see what's going on, I think. Okay, so this molecule is Hiparate. Okay, the sodium is probably associated over here, but I'm not really sure, so I wouldn't really worry about that. Here's sodium hiparate. The significance of this in nature, I really have no idea. This may be just a lab thing that we've realized, hey, this helps us differentiate between our Streptococcus. But our group B streps can make the enzyme hiparicase, which cleaves the hiparate right here. Okay, so what that gives us, we've got a molecule that looks like this. Okay, you see this benzene ring here. Okay, that's we'll call it benzoic acid, or benzoate or sodium benzoate, either way. As well as, let's see if I can fit this in here. Okay, this molecule, 3-carbon, oh, it's hard to see my third carbon in there. A little hard to recognize with my handwriting there, but should recognize that. Here's an amino group. This is one of the amino acids, glycine. Okay, so this is an easy product to test for. We have lots of reagents that turn color in the presence of proteins or amino acids. Okay, so your tube, your sodium hiparate tube, is going to look like this tube here when you inoculate it. It's the same smaller glass tubes that you use to inoculate a coagulase test, but of course there's sodium hiparate media in there this time. Like the coagulase test, though, you want to make sure you get a heavy inoculum. Okay, your book even says to use a swab to inoculate this. I don't generally encourage you to do that because sometimes when you stick a swab in here, it just kind of soaks up all the media. So I would use your loop, but you want to get enough organism on your loop that you can see it. Once you stir it around in here, it should look a little bit cloudy. Okay, so get a nice heavy inoculum. That's important for this to turn out right. And then after incubation, you're going to add a reagent called Ninhydrin. I think it's only 3 drops. Ninhydrin is going to turn purple in the presence of amino acids. Okay, so you'll get a nice purple color change for a positive result. It starts out really light lavender, and then as it sits, it will get darker and darker. But this one does take a little time to react. And like I said, it's not as foolproof a test, I think, because sometimes we just don't get enough organism in there to begin with. So usually it works, but again, not always. Okay, the last test that I want to talk about is a camp test. We typically don't have students inoculate this. We usually just use it as a demonstration. Why don't we have students inoculate this as well? Well, you have enough going on in this lab for one thing, but this is another test for group B streps. So it's a little bit redundant. We already have a group B test, and it's a little bit more difficult to get this to come out right. But let me just talk about the basics here. The camp test is really testing to see if an organism has what's called the camp peptide. And the name here came from the 4 people that developed it. It's not anything to do with cyclic AMP or anything like that. It's a little bit unusual how you inoculate this. You take a blood agar plate and you streak a line of Staph aureus down the center. Okay, so it's unusual that you're purposely putting more than one organism on the plate here. Okay, and then the organisms that you want to see if they have the camp peptide, you streak perpendicular to but not quite touching that line of Staph aureus. So say I streak some Strep agalactiae here, and maybe I want to test another organism like Strep pyogenes. I might put it down here. Again, perpendicular to, but not quite touching that line of Staph aureus. Okay, these 2 organisms are both beta hemolytic, so you may be helpful to try to tell them apart in this way. And then after you incubate, for a positive result, you're looking for an area of increased beta hemolysis or enhanced beta hemolysis. And it's got to be in this arrowhead shape. Okay, so here the Strep agalactiae, you see this nice kind of triangular or arrowhead shaped area of increased beta hemolysis. Okay, sometimes you see, even for a negative result, you might see a little enhanced hemolysis like this, but it's usually just a blob instead of a nice triangle like this. Okay, so, oh, sorry, I had to sneeze. So organisms like Streptococcus, they have this CAMP peptide that works together with hemolysins produced by Staph aureus. Staph aureus is actually beta hemolytic as well, even though we didn't talk about that before. So the CAMP peptide works together with hemolysins produced by Staph aureus to give this increased beta hemolytic effect. Okay, if you want to inoculate a CAMP test, you're welcome to do that, and you're welcome to do it later on for your unknown if you like, but that won't be required of you. Thanks for listening. I know that was a long one.